Our next speaker, Zoya Tawhidi. Yeah, she just went like this. That's okay. Okay, I want you to introduce yourself. Zoya is coming from the University of Toronto, supported by the Canadian Blood Services, the collaboration, and works with Alan Lazarus at U of T. Zoya.
Hi everyone, my name is Zoya Tawhidi, and thank you for the introduction. So I'll be speaking about my project today that looks at mechanisms of platelet destruction in fetal and neonatal alloimmune thrombocytopenia, or FNAIT. So FNAIT, it's a complex pregnancy disorder, and it's caused by maternal alloimmunization, where essentially exposure to fetal platelets that express a paternally inherited antigen, the mother becomes exposed to them and subsequently creates alloantibodies, and these antibodies are IgG, so they can cross the placenta and bind to fetal platelets, subsequently clearing them. This causes the low platelet count or thrombocytopenia that we see in fetuses and neonates. The most commonly reported antibody is against the HPA-1a antigen, but currently the precise pathophysiological mechanisms of how anti-HPA-1a antibodies cause this fetal platelet destruction remain unclear. So we wanted to explore some mechanisms that may be implicated in this platelet destruction. First off are Fc gamma receptors. So Fc gamma receptors are a class of immune cell surface receptors, and they're the primary receptors for IgG and are expressed differentially on immune cells. The majority of Fc gamma receptors are also known to be involved in the activation pathway for macrophages. Next is the complement. So activation of the classical complement pathway could lead to platelet phagocytosis via either complement receptors or even direct platelet lysis. Another possible mechanism of clearance is through platelet activation. So platelets can undergo removal from the bloodstream following activation, which can essentially reshape their morphology and phenotype of platelets. There are common signals for active platelet clearance, including the exposure of key molecules such as P-selectin. And lastly, a mechanism that has not been studied at all yet in the context of FNAIT is platelet desylation. So platelets can self-regulate clearance through endogenous modification of surface glycans. In particular, the process of removing their surface sialic acid. And in certain pathological conditions, this can lead to altered platelet clearance and enhanced clearance. So the objective of this study was to assess the ability of sera from 10 FNAIT patients, so women who've had FNAIT babies, who are all detected to be anti-HPA-1a positive, to mediate Fc gamma receptor-dependent platelet phagocytosis, complement fixation, platelet activation, and platelet desylation, and then to determine if a combination of these mechanisms could explain the thrombocytopenic severity we're seeing in patients. So to assess platelet phagocytosis, we use confocal microscopy, and for the sake of time, I've excluded that, but if you have any questions, it's on my poster. I'll walk you guys through this figure. So we tested 10 patients' sera's ability to trigger phagocytosis using a third-party healthy donor platelets. And so the sera here is organized in ascending order, and the normal human serum plus 2 times the standard deviation is kind of the arbitrary cutoff to determine if a sera is positive or not for triggering phagocytosis. And by our analysis, 9 out of the 10 sera did cause phagocytosis in vitro. The bars are different shades of red depending on the patient's thrombocytopenic severity. So this is interesting because the only patient that didn't trigger phagocytosis was patient J, and as you can see, they were considered severe for having thrombocytopenia. So on that note, we tested the sera's ability to fix complement. And as you can see in the bottom row, patient J was one of the two patients that was able to fix complement. So it kind of shows us that beyond the antibodies being present and causing phagocytosis, there is some role for complement. But with patient B having both phagocytosis and complement, these mechanisms don't necessarily have to be mutually exclusive. There was also patient A that had mild thrombocytopenia and also did trigger phagocytosis. So next we wanted to confirm if there was a role for Fc gamma receptors. So we selected 3 individual FNAIT patient sera, and we blocked Fc gamma receptor 1 and Fc gamma receptor 3, and we found both of them to significantly decrease the level of phagocytosis, showing that both of these receptors are predominantly involved in phagocytosis. We did have some other data with Fc gamma receptor 2, but again, for time, I've excluded it. But based on previous studies, Fc gamma receptor 2 doesn't really seem to have much of a role in platelet phagocytosis using this mechanism. And finally, I want to share with you the data we collected looking at platelet activation and desylation. So we studied platelet activation using CD62P or P-selectin expression and desylation with RCA1, which is essentially a lectin that binds to desylated glycoproteins, including desylated platelets. And so when the patient series were assessed, as you can see, for both activation and desylation, and both of these mechanisms kind of exist together in a positive feedback, you can see that both for patients C and I, they have significantly higher levels of P-selectin expression and platelet desylation in comparison to our resting platelets. This is interesting because both of these patients were also categorized to have severe thrombocytopenia. So this suggests that there's also this more complex pathophysiology that might be underlying this platelet clearance. And so this brings me to the main takeaway point that Fc-mediated phagocytosis and complement seems to be describing the majority of our cohort of patients, but in select patients we're also seeing clearance mechanisms such as desylation and activation. And that concludes my talk. Thank you all for listening, as well as to my supervisor and lab and collaborators.
So if you take platelets and treat them with sialidase, what happens? Do you get upregulation of P-selectin?
Sorry?
If you take platelets and you desylate them, do you get upregulation of P-selectin?
No, so sorry. These tests were done separately, but naturally, like platelets that are desylating are most likely getting cleared through activation as well.
So that suggests that there's an association. I was just asking, is there a cause of one to cause? Is there what's causing the chicken or the egg? What's happening?
Oh, they're two separate mechanisms, so I wouldn't say they're working like together, but we see them happening like at the same time in pathological conditions.
Any other questions? Or you can catch her at our poster. Great. Oh, up there. Yeah.
So this is happening. This is happening in the baby, in the fetus, right? So, is the fetus immune system mature enough for all of these mechanisms? I guess is what I'm wondering.
That's a good question. So there's like for example for desylation, when we look at ITP, which where they have autoantibodies that target the same integrin, it is happening through hepatic clearance, and that is like something we're not sure if that's what's happening with the fetus as well and at what stage. Also with the HPA-1a antigen, alloimmunization can happen very early, even before platelets, because it's expressed on different fetal cells. So that's kind of a question mark.